Sir, please start.
Dr. Rajendra, is this visible? My screen?
Yeah, it is visible. You can share that one. Good. Shall we start?
Yes.
Okay.
Please go ahead.
Okay. Thank you. Yeah. Good evening, all the participants who are viewing us over online through YouTube channel as well as a Google Meet. Good evening once again to all. Today we have Professor Som Subra Chakravarti, working as assistant professor in Agriculture and Food Engineering Department. He is going to deliver a lecture on importance of soil testing for crop production. Very informative lecture you can have today by Professor Som Subra Chakravarti. He has more than 100 publications related to the soil science and soil science and hyperspectral proximal soil sensors, portable XRF applications in soil, digital soil mapping. These are all the different areas he has worked. He has published more than 100 publications in these areas. You can interact with him at the end of the session. Thank you. Thank you, Professor Som Subra Chakravarti, for accepting our invitation to deliver a lecture on importance of soil testing for crop production. You can take the session. Thank you.
Okay. Thank you, Professor Machavaram, and good afternoon to all of you. So friends, I'm, as Professor Machavaram has already told you, I'm working as an assistant professor in Agriculture and Food Engineering Department. Let me make my presentation in the slide view mode. So I hope it is now visible to all of you. If you have any questions and queries, please note it down and you can ask me at the end of my presentation. Okay. So today we are going to discuss a very important topic, that is importance of soil testing for the crop production. Now please understand that this soil testing is a vast subject and it requires several years of knowledge and practice for make yourself very much conversant with different technologies of soil testing. So in today's lecture, since our time is limited, I will try to give you a brief overview of traditional soil testing. Obviously, at Agriculture and Food Engineering Department of IIT Kharagpur, we also do advanced soil testing using some advanced methodologies. If time permits, I'll give you a glance of those technologies, but my major intention for this training session is to give you an understanding, basic understanding of what is soil testing and how we do soil testing, what are the steps of soil testing, and if you want to develop a soil testing lab, how you should proceed and what should be the financial involvement in that. Okay. So let us start with the basic definition of soil testing. So what is soil testing? Now, soil testing is an acceptably accurate and rapid soil chemical analysis for assessing available nutrient status for making fertilizer recommendation. As you can see, a typical soil testing laboratory in India. So it is basically a method of, it is basically a combination of methods, soil testing. And it is a rapid soil chemical analysis for assessing available nutrient. Now, to understand what is available nutrient, you know that plants grow in soil and soil contains different types of nutrients for the growth of the plants, and get those nutrients from the soil through their roots. Okay. So the fraction, for example, nitrogen is an important plant nutrient. Okay, so plants require nitrogen for their growth. Without nitrogen, they cannot grow, right? Now, nitrogen can be present in soil in different forms. As you know, it can be present in protein form, which is an organic form. It can be also present in inorganic forms like ammonium, like nitrate form. Now, the available nutrient is that fraction of a nutrient which is available to the plant. Now, remember that it doesn't matter in different, if soil contains different forms of nitrogen, plant can only take nitrogen in some certain specific forms. For example, plant can only take nitrogen in ammonium and nitrate form. It doesn't matter whether other forms of nitrogen are present in the soil. So generally, when we talk about soil testing, we focus on this available nutrient content. Okay. Again, available nutrient content is that fraction of the total nutrient which is available to the plant. That means plant can only take that nutrient and plant cannot extract any other forms of that nutrient. For example, in case of soil, you know that nitrogen is present mostly in organic form, but plant cannot take organic forms of nitrogen. So measuring the total nitrogen in the soil doesn't have any meaning. So we should measure only the nitrate and ammonium which plant can take. So that is called available nutrient status. So in the soil testing, generally we measure available nutrient content in the soil to make fertilizer recommendation. Just like when you have some disease and you go to doctors, and doctor, before prescribing some medication, he gives suggestion for some testings like x-rays, like MRIs and other types of testings like blood test. So you can understand that soil testing is a similar type of approach where we do different types of chemical analysis of this available nutrient content in the soil. Okay, and from there we try to have an idea about soil fertility status, and then we make fertilizer recommendation. Okay, so this is about the soil testing. Okay, now to understand the soil testing, let us see what are the major components of a soil. So soil is basically composed of three phases: solid phase, liquid phase, and gaseous phase. Okay, now the solid phase contains 50% of the soil volume, and the pore space, and the rest 50% is basically pore space. So as you can see here, the 50% of the solid space is occupied by 45% mineral matter and only 5% organic matter, and rest 50% pore space is occupied by air and water. And here you can see that air and water are interchangeable, where water content more, air will be less, and vice versa. So you can see in a soil, it contains both solid phase, atmosphere, and also soil water. So it is a basically 3 to 4 phase system. Okay. Now, in this mineral matter, soil contains different types of nutrients, and these nutrients are basically taken up by the plant for their growth. Just like human being, we are dependent on some food to sustain our life. Similarly, in case of plants also, they require food, and these foods are these nutrients which they get from the soil, and these nutrients are basically present in this mineral 45% mineral fraction. Okay, now let us see what are the essential nutrients. Now you see that in the soil it contains hundreds of different elements, but this term is very important. What is plant essential element? Essential elements are those elements which are indispensable for the growth of the plant. That means plant cannot survive or cannot complete the life cycle without the presence of this any of these nutrients. So there are 17 total plant nutrients. Okay, so there are 17 essential plant nutrients. Just like in our body, we have essential amino acids. Similarly, in case of plants also, they have 17 essential plant nutrients. So they need to have these nutrients uptaken by them from the soil or from other sources to sustain their life. Now, these essential plant nutrients are again divided into two major categories. One is called framework elements, another is called mineral elements. Now, the framework elements are basically three. What are those? Carbon, hydrogen, and oxygen. And mineral elements are basically 14. Okay, and these mineral elements, see these framework elements like carbon, hydrogen, oxygen, plant basically take from air and water during the photosynthesis process, and the rest 14 elements they take from soil in ionic forms. So these 14 elements again divided into macronutrients and micronutrients. Now the macronutrients, there are 6 macronutrients and there are 8 micronutrients. Okay. So what is the difference between macronutrient and micronutrient? See, both macronutrients and micronutrients are essential, but macronutrients are required in higher quantity and micronutrients are required in lower quantity. Okay, micronutrients are required in less than 1 ppm concentration to maintain their growth, and macronutrients are required in larger quantity for maintaining their growth. But irrespective of it is macronutrient or micronutrient, they are required because see, you can see that zinc is a micronutrient. So irrespective of the low requirement of zinc for plant growth, if zinc is absent in the soil, plant cannot grow. So this is essential. So you see that we are dividing these mineral elements into macro and micro based on the plant requirement. Okay. Now these 6 macronutrients are further divided into primary nutrients and secondary nutrients. Okay. So there are 3 primary nutrients like nitrogen, phosphorus, potassium, and secondary nutrients are calcium, magnesium, and sulfur. So you can see that the difference between the primary nutrients and secondary nutrients are, so primary nutrients are required in larger quantity than secondary nutrients. Okay. So nitrogen, phosphorus, potassium required in larger quantity than calcium, magnesium, and sulfur. Okay. And another division is micronutrients: zinc, copper, manganese, iron, boron, molybdenum, chlorine, and nickel. So you can see these 8 are different micronutrients. So it is very essential to remember these difference between macronutrients and micronutrients. Okay. And in a typical soil testing lab, we generally measure both primary nutrients, secondary nutrients, and micronutrients. Okay. Now, the macronutrients are basically nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. Now, NPK are often referred to as the primary nutrients, as we have already seen, and are the most common elements found in commercial fertilizers. So you can see most of the time the commercial fertilizer contains these 3 elements like urea, like superphosphate, like muriate of phosphate, muriate of potash. So all these are major fertilizer which generally farmers are utilizing in their field. So they contain these primary nutrients, and that is indicative of higher nutrient requirement for these primary nutrients, NPK. Now the secondary nutrients are calcium, magnesium, sulfur. They are also major nutrients, but they require in relatively less quantity than primary nutrients, and they are also found in fertilizer and soil amendments. So some fertilizer, they supply, apart from primary nutrients, they also supply some secondary nutrients also. They are moderately required by the plant and they produce deficiency symptoms in patches. And generally they are applied through fertilizer containing primary nutrients like single superphosphate or superphosphate. Okay. So now we know what are the micronutrients. Micronutrients are 8 micronutrients: iron, manganese, zinc, boron, copper, chloride, molybdenum, and nickel. And micronutrients are required in relatively minute quantities. However, irrespective of a nutrient, macronutrient or micronutrient, they are indispensable, as I have already told you. Okay. Now, if soil contains less amount of that particular nutrient, the plant will give you some deficiency symptoms. Just like if we have some fever, if we have fever, our how we indicate our temperature, our body temperature increased, right? So similarly, when the soil is deficient in particular nutrient, these plants show different types of deficiency symptoms. So you can see here in this slide, I have presented some deficiency symptoms of nitrogen, phosphorus, potassium. So in case of nitrogen, you will see there are stunted growth. So the plant will not grow and it will become yellow if there is nitrogen deficiency in the soil. So that will indicate that you need to apply more nitrogen into the soil so that plant can get. When the phosphorus is deficient in the soil, you will see there is a stunted growth and there you will see reddish discoloration. So this is an indication of phosphorus deficiency. So whenever you see this type of symptoms, you are sure that the soil needs more phosphorus fertilizer. And for potassium, you can see that there are chlorosis or necrosis of leaf edges. So you can see leaf edge, they become necrotic tissues. They become, wilted. So you can see this is a typical symptom of potassium. So all these symptoms indicate that you should apply these fertilizers of nitrogen, phosphorus, and potassium. Similarly, in case of calcium deficiency, you can see some physiological disorder. Like here, you can see, a disorder of tomato, and here you can see magnesium problem, magnesium deficiency, and here you can see sulfur deficiency in case of crop. All these different deficiency will show deficiency symptoms. Okay. Now, similarly for iron, you will see intervenal chlorosis. In case of zinc, you get stunted growth. In case of copper, you will see this type of problem. And in case of manganese deficiency, you will see this type of problem. So one thing is sure that if a nutrient is deficient in the soil, plant will give some kind of indication that the soil is deficient. And generally, it can be visible. And when you see this type of symptoms, you need to analyze your soil to see whether actually there is a deficiency of that particular nutrient or not. So, when we identify this type of problems, then it is recommended that you should go for soil testing of your farm. Because unless you test your soil, how do you know that the soil contains these nutrients in sufficient quantity or deficient quantity? Okay, so let us go ahead and see what are the objectives of soil testing. Now we know why we do soil testing. Let us see what are the major objectives of soil testing. Now the major objectives of soil testing is to estimate the available nutrient status like macro, secondary and micronutrients of soils. The second objective is to assess the type and extent of problem for reclamation of problematic soil. And thirdly, to evaluate the fertility status of soil of a country or state or a district. So again, we want to measure the available nutrient status of macro, secondary and micronutrients. And we want to assess the type and extent of problem for reclamation of problematic soil. For example, if your soil is saline soil, how do you rectify that soil? Because if your soil is contains salt, plant will not grow, right? So how to rectify that? So all these suggestions will be given by the soil testing expert. And finally, to evaluate the fertility status of the soils of a country or a state or a district. Okay, so these three are major objectives of soil testing. Now let us see what are the utilities of soil testing. What do you want to actually use the results of soil testing? First of all, the soil test summarizes the fertility status, that is available nitrogen status or available phosphorus status or available potassium status, expressed as high, medium or low. So when you do soil testing, it will tell you that for a particular nutrient, whether your soil is contain that nutrient in a high quantity, medium quantity or low quantity. If the soil is contain soil contain low amount of that particular nutrient, you must add some fertilizer. So a soil fertility map showing such fertility status can be prepared, and the soil fertility map can be used for four major purposes. First, to delineating the areas of nutrients like nitrogen, phosphorus, potassium sufficiency or of deficiency. Secondly, studying soil fertility changing pattern due to crop cultivation over a period of years. So when you do cultivation of a particular type of crop, after certain years, you will see your soil fertility has changed. So to study this fertility changing pattern, you need to go for soil testing. Thirdly, to determine nutrient requirement for the deficient areas. And finally, to prepare a basis for fertilizer recommendation. Unless you have, for example, in case of doctors, unless they have blood test report, how would they give you medication? So similarly, for a soil scientist, unless we know the soil testing result, we cannot give the recommendation or for fertilizers to the farmer. So these are the utilities of the soil testing. Now, we are going to discuss the very important point, that is steps of soil testing. Okay. So soil testing basically consists of 5 major steps. The first step is soil sampling. Second step is preparation of soil sample. Third step is extraction or analysis of available nutrients by an appropriate laboratory method. Fourth one is interpretation of soil analysis data. And fifth one is fertilizer recommendation. Okay. So again, first, soil sampling. So it is a very important step. Secondly, preparation of soil sample. Thirdly, extraction or analysis of available nutrients by an appropriate laboratory method. Fourth one is interpretation of soil analysis data. And fifth one is fertilizer recommendation. Until you do fertilizer recommendation, your soil testing is not completed. Okay. So let us discuss them one by one. Okay. So soil sampling. See, when you send our soil samples to the lab for analysis, the collection of soil sample poses the major risk, because if we collect the soil samples in a wrong way, that will give us wrong results which are not useful. So we must make sure that our soil sampling is flawless. That means it is absolutely correct. Okay. So soil sampling is the most important step. Okay. So what are the different types of tools we generally use for
soil sample?
So soil sampling, basically we do soil test. We do soil sampling by using some tools. What are those tools? You know these tools like khurpi, auger, spade, post hole diggers, hydraulic core sampler. So you can see here, these are the spade. You can use the spade to dig the soil samples. And there is a specialized equipment called auger. We use auger to collect the soil samples, okay, from the soil. There are different types of augers, okay, screw type auger, tube auger, post hole auger, different types of augers. And these different types of augers are suitable for different types of soil, okay. So we generally use these types of tools to collect the soil samples. Now the question arises, okay sir, how many soil samples I should collect, first of all, and how to collect the soil samples, okay. So the first answer is, generally from a parcel of land, we generally recommend at least you collect 20 soil samples per hectare, okay. And you must not collect the soil samples from recently fertilized plot. So if you are collecting soil samples from a recently fertilized plot, obviously that soil will have high nutrient content and it will give you wrong results, right? You also should not collect samples from ponds, channels, marshy tracts, near area trees, for example, wells, compost pile, water areas. So these areas should not be sampled. you should collect the samples properly from the field. Otherwise, your soil testing results will be erroneous. Remember, in case of blood sugar testing, we have to follow certain rules. For example, we have to take, without taking any food and after taking some food, okay? So we have to maintain that norm before we give our blood sample for blood sugar testing, right? So similarly, for soil testing also, we should be very, very careful to collect only those representative samples which will give you the correct results. Okay. Now the next question comes, how to collect the soil sample? Your field is vast. So how to collect the soil samples? The most recommended method for collecting soil samples is the zigzag way. As you can see in this picture, the soils are collected from these black dots. So you can see these black dots are basically arranged in the field in a zigzag way. Okay. So in zigzag way, we should collect 20 soil samples per field. Now, why zigzag way? Because when you do sample in this fashion, it will cover most of the areas of the field. Okay. Instead of if we just take a random sample from any spot, if we do this kind of sample collection, you can see that you are basically covering the whole field. Okay. And once you collect these 20 soil samples, you mix them together to form a composite sample. We call it composite sample. We mix all these 20 soil samples collected from 20 spots in a single bucket to make a composite sample. Okay. So there are other methods of soil sample collection. As you can see, this is a random sampling. Sometimes you will see that there are some random sampling without following any pattern. So B is the zigzag method. I have already showed you the zigzag method in our last slide. And another method is there that is called subsampling a large field. So in these first two methods, like random and zigzag method, these fields are up to 1 hectare of area. So this C type, that is subsampling a large field, is recommended when your field size is quite big, that is 15 to 16 hectare of area, with each spot indicating the site of a subsample. So you can see that we are collecting first subsample here. So 5 samples we are collecting and then we are mixing them together. Then again 5 samples we are collecting, then mixing them together. So this is how we do the subsampling a large field up to 15 to 16 hectare of area. Okay. Now once we collect the samples, we mix them to composite samples. So generally we mix these 20 soil samples together to develop a composite sample. Okay. And so we have to prepare a composite sample from each uniform field. Now you will see some strange things when you go for soil sampling. You will see that some parts of your field are basically organic in nature. Some parts contain high amount of sand, and some parts contain some high amount of clay content. So you should collect the representative composite samples from each 3 parts. So 20 samples from clay in a zigzag fashion and mix them to make a composite sample. Again 20 samples in a zigzag way from the sandy part and then mix it in a composite sample. And finally, from the muck or organic matter, also you collect 20 samples zigzag way and then mix them together. Similarly, if we see that some portion of the field has been limed first 5 years ago, and some portion of the field has not been limed, so you should collect samples from both the areas. Okay. Now, you can see here, formerly 2 different fields, now all in a single field. So you should collect also 2 composite samples. Okay. So, you are applying manure, organic manure in one part of the field, and you are not applying any manure in another part of the field. So you should collect 2 samples from these 2 composite samples. So also you can see that well-drained soil and then steep slip soil and flat flat soil, there is some upland soil. So from those areas also, you should collect 3 composite samples. Okay, and also you can collect in a big field, you can uniformly divide them, and also you can collect some composite samples. So this is how you should collect these composite samples. Okay. Now, what depth you should collect the samples? Okay. For most of the cereals or vegetables or seasonal crops, we generally collect the samples from 0 to 15 centimeter of the soil. Okay. From the top, 0 to 15 centimeter, we go up to 15 centimeter, and we call it furrow slice layer. Why we go up to 15 centimeter? Because for most of these crops, their roots will go up to 15 centimeter. They will not go beyond 15 centimeter. So whatever nutrient they will uptake, it will be basically from this 0 to 15 centimeter. So that is why we should focus on this depth of sampling of 0 to 15 centimeter. Okay. However, fruit crops, you can collect the samples from 0 to 30 centimeter, 30 to 60 centimeter, 60 to 90 centimeter, and also 4 or 5 pits dug in 0.5 hectare area. In case of saline alkaline soil, you can collect it from 0 to 15 centimeter. In case of grassland, if your soil is basically comes under the grassland, you should collect it from 5 centimeter, because for the roots of the grass, it should not go beyond 5 centimeter. So you see, depending on your crop, you should collect the samples, but most of the time, we generally collect the samples from 0 to 15 centimeter for soil testing purposes. Now, how to collect the soil sample? I told you that we use auger. So here you can see a specialized auger, which we generally push into the soil, and it will take out a chunk of soil from a desired depth. So this is how we use our soil, use auger to collect the soil samples. Another method of collecting soil sample is you make a V-shaped cut into the soil and discard all the materials within this V. So after you cut this V-shaped cut, you take out 1/2 inch or 1 inch of slice from both the sides and then mix them together to get your soil samples. So this is how you also can collect the soil samples. We call it V-shape cutting. And after you cut this V-shape, you collect 1/2 inch thick slice from both the sides and mix them together to get your total samples. Okay. So as you can see here, he is making a V-shaped cut to a depth of 0 to 15 centimeter in the sampling spot using spade. And also here they are using this auger to a plow depth of 15 centimeter. This is called also plow layer or plow depth, and draw the soil samples, just like I have showed you. Okay. So these are 2 ways to collect the soil samples. Okay. Now, what are the procedure of soil sampling and processing? Okay. So the first step of soil samples is when you go to the field, you remove some surface litters, grass, debris, etc. from the sampling point. So from the point from which you want to take the samples, you remove those grasses, surface litter, debris, and then you drive the auger to a plow depth of 15 centimeter and draw the soil samples. And you should collect 20 samples from each sampling unit and place in a bucket or tray. As you can see, I told you about this 1/2 inch to 1 inch slice from the V cut. Okay. And you can see there are different types of auger, screw auger, then tube auger. Okay, different types of augers are there. So if auger is available, excuse me. So if auger is not available, make a V-shaped cut, just like I have showed you in the last slide, to a depth of 15 centimeter in the sampling spot using the spade, and remove the thick slice of the soil from top to bottom of the exposed face, that is 2.5, that is 1 inch, half inch to 1 inch of the V-shaped cut, and place them in a clean container. And collect the soil at one place, mix thoroughly with hands, and spread on a paper or clean plastic sheet and make 4 quarters. So one thing is, once you collect the soil samples, you dry it and grind it, and then you can spread on a paper or clean plastic sheet to make 4 quarters, just like here. As you can see, we are spreading the soil samples and making 4 quarters. And then the next step, we are removing the opposite 2 quarters and return the other 2. So we repeat the same process until we get total of 500 grams of soil. So 500 grams of soil is sufficient for doing the soil testing. Okay. Remember, you should dry the soil always in shade. Never ever dry the soil samples in the sunlight. Because if you dry them in the sunlight, some of the nutrients will get changed into another form. Some of the nutrients will convert and you will not get the accurate result. Because when you keep them in the sunlight, the heat of the sunlight makes some chemical changes in the soil, and as a result, some nutrients will change and you will not get the correct results. For example, the organic carbon is an important parameter of soil testing. So if you keep the soil in the open sunlight, the organic carbon will be oxidized to carbon dioxide. So you will not get the correct result. Okay. So once you dry the soil samples in the shade, you powder them by crushing them, and then you sieve it by a 2 millimeter sieve. Remember that. The 2 millimeter sieving is very, very important, because whatever soil testing we do, it generally, we generally do it with the fraction which is less than 2 millimeter. Anything which is beyond 2 millimeter, we cannot term that as soil. Okay. And then we put the sample in a plastic bag, label it properly, and submit it for the analysis. So this slide shows different steps of sample collection. As you can see, we are first removing the surface litter, and then sample collected by V-cut, and then we are doing the drying and sieving. So drying generally done in the shade, okay, and in the air, and after you dry them, you crush them and you sieve them using 2 millimeter sieve, and then you divide them into 4 compartments. You do the quartering, or opposite 2 quarters discarded, and finally you pack them in the sample bags and properly label it. Okay. Now you can ask me, okay sir, how to grind the soil samples and sieve it? So for grinding, there are different types of methods. Generally, some mechanical grinders are available in the market. So if we use them, the grinding process becomes very, very easy. Otherwise, we have to use some mortar and pestle to grind the soil samples. After we crush and grind the soil samples, we have to sieve them. And as you can see, we are sieving them to discard these roots and other foreign particles, so that only 2 millimeter, less than 2 millimeter size soil samples get retained, and this will be required for future analysis of soil testing. Okay. So this slide shows the step-by-step method of soil sample collection and its processing. So when we go to the field, the soil is moist, so we collect them in large quantity. Suppose we are collecting in the composite sample contains greater than 500 grams. We describe the samples, and then we dry them in the air, and sometimes we dry them in the oven, and sometimes we dry them in the freeze dryer. So I'm not going to cover why we dry them in the oven and freeze dryer, but they are for certain applications. Generally, for soil testing purpose, we prefer to dry in the air, in the shade. And after we dry them in the air, we remove the gravel, glass, roots, etc. by sieving. Okay, so we sieve with a 2 mm sieve and we get fractions less than 2 mm. Okay, so after we get the fractions less than 2 mm, then we do the quartering and we're subdividing the samples to keep only 500 grams of samples. And this quartering method will ensure that you are collecting only 500 grams of sample, because it is of no use to collect several kgs of a single sample. Otherwise, you will have problem with your storing. So 500 grams of sample will be sufficient for any soil testing purposes. So 500 grams. So our final aim is to keep it up to 500 grams. Then after we collect them, these samples, we can go for further crushing of the samples for less than 250 micron, but this is generally not recommended. This is generally not required for traditional soil testing purposes. So if we have less than 2 mm sieved sample, that will be fine. Okay, so this flow chart shows step-by-step method of soil sample collection and their processing. Okay. Remember a couple of things. The best time of sampling is before sowing or planting. So each sample should have a label describing the field identification, farmer's name and address, previous crops, and the crop for which nutrient recommendation is sought. And for soil sampling, special augers with a core diameter of 1 to 2 centimeter are convenient, but small spade, I have already showed you how we are using spade. Now, another important thing you can use while sampling, you can use the GPS. I know some of you have already heard about this instrument called GPS. Anyway, everyone's smartphone is now contain this GPS, and this GPS is the short form of Global Positioning System. So using this GPS, you can pinpoint the location of any area over the Earth's surface. So if you are collecting a sample from a particular spot, if you get the GPS coordinate of the spot, you can always go back to the spot after a couple of years and see whether the soil fertility status has changed or not. Also, these GPS points are useful for making the map of fertility of a particular area. So these type of things are very, very useful. And earlier we used these, GPS instruments for research purpose. However, nowadays everyone has their smartphone, and in the smartphone there is a GPS which you can use to get the location of a particular soil sampling spot. Okay, now once we collect the soil sample, you process them and you are packing them in the bag. The bag should contain several information. What are these information? These information are absolutely necessary for soil testing. Okay, because if you miss this information, the final recommendation cannot be generated. First of all, name and address of the farmer or the farm owner, name of the block, plot number or any other number that identifies the plot, soil texture, whether it is sandy, clay and loamy type of soil, whether there is an availability of irrigation facilities, whether there is an availability of drainage system, whether the land is upland, medium land and lowland, and what is the depth of soil sample. So these information must be there in the bag of soil testing. And also, these information also should be kept. For example, information about the previous crop, like name and variety of the crop, dose of organic manure if applied, dose of fertilizer if applied, yield of that previous crop, and also information of the crop that will be grown, like name and variety of the crop, which season you want to grow that crop. If you are facing some problems, what is the date of sample collection? So all this information must be there in the bag of sample. And finally, there should be signature of the farmers or farm owner. So these are the information that are required for making a perfect soil testing recommendation. Now, you can ask me, okay, sir, we have now understand about the basics of soil testing. and why we do the soil sampling. Now, the next question comes, what should be the frequency of sampling? Now, remember that for most field crop applications or field cropping systems, sampling and testing of soils in each, for each field at least once in every 3 years is adequate. So generally, it is recommended that you should test your soil sample once in a 3 year. Okay. And another important point is sampling the entire farm at one time is a good practice because it provides an evaluation of the whole farm fertility program at a given point of time. However, sometimes it is not practical if your farm is quite large. So for large farm operation, sampling and testing, 1/3 of the acreage each year is an alternative that provides continuity over the time. So basically, if you have, for example, if you have 3 hectares of land, so each year you should go ahead with 1 hectare of land soil testing. Okay. For intensive cropping system where large amount of fertilizers may be applied annually or crop removal may be high, annual soil testing enables the grower to maintain stable soil fertility conditions, and this is especially important for many of the vegetable crops that are grown on sandy soils. Okay. Now, in a typical soil testing lab, what are the parameters generally we measure? So these are the parameters which we generally measure in the soil testing lab. We first measure soil pH, then soil EC or electrical conductivity, then we measure soil organic carbon or SOC, then we measure available nitrogen, phosphorus, potassium. I have highlighted them, or I have made these blue just to indicate that the smaller soil testing laboratories where the resources are limited, they are generally giving us generally give the recommendation for these 6 parameters like soil pH, electrical conductivity, soil organic carbon, available NPK. Okay. However, for a full-fledged soil testing laboratory, they also measure available calcium, magnesium, and sulfur, and also they measure available micronutrients. Okay. So we are going to discuss each of these parameters and how we generally measure them and what are the basis of recommendation we'll also discuss. Okay. Now we remember one thing, that for estimating these nutrients we need to have certain kind of chemical reagent. We call them extractant. Why we call them extractant? Because these extractants are, I told you that we are not measuring the total nitrogen, total phosphorus, total potassium, or total any other micronutrients. We are measuring available forms. So to extract only the available form, we need to have some specific chemicals. So these specific chemicals are known as the extractant. Okay. So for example, in case of available nitrogen, the extractant we use is called alkaline potassium permanganate, and the instrument which we use is called Gendal apparatus. And for phosphorus, available phosphorus, we generally use 2 methods. One is called the Olsen's method, another is called the Bray and Kurtz #1 method. So Olsen's method is generally used for neutral or alkaline soil, whereas in case of Bray and Kurtz method, this is meant for acidic soil. Okay, and generally we use spectrophotometer to measure this available phosphorus. We'll be discussing the details of spectrophotometer in our coming slides. Available potassium we measure by ammonium acetate and extract by ammonium acetate and measure through flame photometer. For micronutrient like zinc, copper, manganese, and iron, we use a specialized extractant called DTPA The full name is diethylene triamine pentacetic acid. And for boron, we use hot water to extract the available boron. And for molybdenum, we use Griggs reagent, that is ammonium oxalate of pH 3. See, there are several other also methods, but I have here mentioned the most important one which we follow in India. There are also other methods which are being used not only in India but also in Western countries. But I have given here only those which are well established in all the laboratories. Okay, so let us, if you have any question, you can ask me at this point of time. Whatever I have covered so far, based on whatever I have covered so far, if you have any question, you can ask, and then I'll start the next phase. Okay. Do you guys have any questions so far? If you have any question, you can ask me, and I'll be trying to give you answer, and then I'll start the next phase.
Sir, this is Ram. You mentioned the red hot water we can use it for boron extraction, right? It seems a little easier to do that. Is it possible to do in the farm itself?
In the farm?
Yeah.
Yes, you can do it in the farm also, but the instruments which we are using for measuring that is not portable. So the hot water extractable thing has to be, these are basically standardized laboratory technique. So the other instruments, other tools which you require for the measurement is basically, generally laboratory setup. So it is generally recommended to do in the lab. Okay.
Okay, sir. Thank you.
Any other questions?
Soil organic carbon is measured. Just want to understand the high level process.
Yes, I'll discuss them the process in the next phase. Okay, but if anybody has any question regarding part which I have just covered, you can ask me. Okay.
Sir, one more question. Is there any reference card, right, to find out the range, right, which is medium, high, low? Is there any reference card for each of these micro and macronutrients?
Obviously, there are some reference card for these micro. I will show you the reference card for these for nitrogen, phosphorus, and potassium. Okay, and for micronutrients also there are some sufficient, deficient, high, medium, low levels. Remember one thing: these high, medium, low nutrient content are also region specific. So the cutoff values for high, medium, or low content for West Bengal may not be the exactly same with that of Tamil Nadu. Okay, so they are having their own, these values also. Okay, so these values may change, but I'll show you some, some universal. Some of them are more or less accepted. I will show them. Okay. Okay, I hope that you do not have any, there is no further question. Okay. Yeah.
Hello, sir.
Yes.
Yeah, Vanita Bala here, sir. I have a doubt. So, if the area is very big, like 5 acres and 10 acres and all, then how many samples, assisted, the quality of soil differs from place to place. So, if that is the condition, so how many samples I need to take it, sir?
I have already showed you in case of that, for if the soil is quite big, then we'll be going for subsampling. See, all this depends on your resources. Okay. Always the better practice is to take as many a sample as possible. But generally, I would recommend that if your soil has some variation, if the field is too big and you understand that some parts of the field are highland, some parts are lowland. So I would recommend that you collect 20 samples from the highland and then 20 samples from the lowland, make a composite for the highland, make a composite for the lowland, or you can do more composite samples also. Okay, so it is always better to have more sample instead of less samples. But why we are not saying that you should collect hundreds of composite samples? Because as the sample number increases, their cost of estimation also increases. Okay, so you have to make a balance based on your requirement and based on your resources. If you have huge resources, if you have no binding in terms of monetary involvement, you can collect as many sample as possible. Okay.
Okay, sir. Thank you, sir.
All right. I hope there are certain questions there. People ask that which one best for hill area soil test like Tripura? Which one best for soil test? Suggest some cost-effective tools for soil testing. Yes, I will be discussing those cost-effective tools for soil testing. Appreciate if you can put the slide in full screen mode. Okay. Okay. Is there any good mobile app for scanning and identifying nutrient deficiency more reliably? Finding it difficult to differentiate between nutrient deficiency and disease. Yes, please give this business for help. Okay. Okay. So I'll discuss these all these, but I do not understand the last question. Which one is the best to hill areas of soil test? If you can clarify your question more, I'll be I'll be trying to give you the answer. Suggest some cost-effective methods. Okay, I'll be discussing that. Okay, now let me go back to I hope this slide is visible to all of you. This slide is visible?
Yes, sir. Yes, sir.
Okay. 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 So different extractants are used to extract a specific available nutrients. For example, in case of nitrogen, we for in case of nitrogen, we are using the alkaline potassium permanganate method, using Zendal method. In case of phosphorus, we use available phosphorus using Olsen method or Brain-Kurtz #1 method. Available potassium is measured through ammonium acetate method using flame photometer. Micronutrient, we use DTPA method using atomic absorption spectrophotometer. I'll be discussing their principle and how they operate. Boron using hot water and molybdenum using Griggs reagent. Okay, so let us start with the soil pH. Now, you all know that soil pH basically indicates whether your soil is acidic or alkaline or neutral. So soil pH basically ranges from 0 to 14, and the soil is neutral when the pH is 7. But the soil, when the pH of the soil is greater than 7, then we call it alkaline soil, and when the pH of the soil is less than 7, then we call it acidic soil. Remember, for most of the plant nutrients are available in the neutral pH range. When the pH of the soil is varying from 6.5 to around 7.5, then most of the nutrients are available to the plant except for 1 or 2. Okay, so it is essential to maintain the soil pH in the near neutral range. Okay, so that is why the first parameter we measure in any soil testing lab is the soil pH. Soil pH is called the master property because soil pH determines the availability of all the other nutrients. Excuse me. So that is why the first parameter we should measure is the soil pH. Okay. Now, soil pH is generally measured in the laboratory using the pH meter, and the principle of pH meter is the pH meter has 2 electrodes. One is called the reference electrode and another is called the standard electrode. So the pH meter is submerged into the soil water solution. And what happens? the differences of H plus concentration between soil suspension and glass electrode gives rise to an electrometric potential. So when we dip this, so there is a glass bulb which is made of a very thin glass layer. So when we dip this electrode into the soil's soil water solution, the H plus ions which are present in the soil, they will produce a potential between the outer membrane as well as the reference electrode, which is present inside the pH meter. And this potential is measured and converted into pH units. So you dip this pH probe and it will give you the results of pH within a couple of seconds. Now, what are the steps in measuring the pH? Now, pH meter is first calibrated using a known pH solution. Generally, we use 3 buffer solution like pH 4, pH 7, pH 9.2. And then we mix 10 gram of soil with 25 ml of water. Okay. And so when we mix 10 grams of soil with 25 ml of water, it will give us 1:2.5 soil water ratio. And from there, then we mix well with a glass rod and allow to stand for 30 minutes. We steer the suspension every 10 minutes during the period. And after 1 hour, we steer the suspension and take the reading. And also, since pH is dependent on environmental temperature, if there is a change in temperature, that should be also taken care of, and the temperature correction must be done. Okay. So when we get the pH results, the pH results also can be interpreted using these values. So if your pH result is less than 4.5, that means your soil is extremely acidic. If your pH is between 4.5 to 5, then it is very strongly acidic. Then you can see the other classes like strongly acidic, mediumly acidic, slightly acidic, neutral, mildly alkaline, strongly alkaline, and very strongly alkaline. Remember, most of the plant nutrients are available in the slightly acidic to neutral condition. So we should maintain this near neutral condition for better extraction by the plant. Okay. Now you see, this is a pH meter. This is a thermoscientific Orion Star pH benchtop pH meter, which we generally use for measurement of pH. There are also other models made by other companies like HANA, like other like other companies also. So which we use for measuring the soil pH. The next important soil property is called the electrical conductivity. Now, electrical conductivity is the capacity of soil to conduct the electricity. Now, since ions contributes to electrical conductivity, so why we measure electrical conductivity? Electrical conductivity is generally measured to measure the salt content, because if your soil is contain high amount of salt, the plant will not grow in that soil. Okay. So to measure the salt concentration in your soil, you need to measure the electrical conductivity. And why electrical conductivity? Because salts are basically made up of ions, and ions conduct electricity. So if we can measure the electrical conductivity, that will give you an idea about the salt content of the soil. So using electrical conductivity, we measure the salt concentration. What are the steps? So you can see conducticity meter, we use conductivity meter. So we first calibrate the conductivity meter using a known standard solution of 0.1 molar KCl. Then we use 10 grams of soil, which is mixed with 20 ml of water, 1:2 soil water ratio. The suspension is then mixed occasionally for 30 minutes. Then conductivity meter is immersed, just like pH meter, we immerse the conductivity meter, and then we take the reading, and then we do the temperature correction just like in case of pH. So what are the interpretation based on the conductivity? So electrical conductivity can be divided into 2 major classes. If the electrical conductivity is greater than 4, this is decisiemens per meter, then we call it a saline soil. So saline soil will have an electrical conductivity of greater than 4 decisiemens per meter. And non-saline soil will have an electrical conductivity of less than 4 decisiemens per meter. Okay. So you can see in this picture there is a saline soil, and in this saline soil no crop is growing. Okay. So if the soil is saline, no crop will be grown. So you must have a non-saline soil for the crop growth. Okay. So what happens if your soil is basically if your soil comes very saline, what you should do? The best way to rectify this problem is to apply non-saline irrigation water in frequent doses or frequency, in high frequency. That means you should apply irrigation water. That irrigation water has to be salt free. Okay, so you should apply irrigation water, salt free irrigation water, small amount in frequent interval. So what will happen? This salt will go down after mixing it with the irrigation water, and it will ultimately leach down into the groundwater. So this is how you can control the electrical conductivity. Also, you should apply organic matter into the field, like farmyard manure, like cow dung, like compost. If you apply compost or any other organic manure, what will happen? The acids will come out from those organic matter after decomposition, and they will dissolve these salts, so to rectify this problem. Okay, what happens if your soil is acidic? So if your soil is acidic, you should ask your farmers to apply more lime, because lime, when you apply lime, that will neutralize the acidity. Okay, and if your soil, after doing the pH measurement, if your soil is alkaline, then you should ask your farmers to apply gypsum, because gypsum is basically calcium sulfate. So when we apply calcium sulfate, that calcium sulfate will neutralize this alkalinity, and it will try to bring down the soil pH. So I told you what should be your recommendation when you have acidic pH, when you have alkaline pH, and what you have when you have saline soil. Okay, so these are some general recommendation. Unless you get this number, you cannot tell them what to do. Okay, so this is why we do the soil testing. Okay, so this is the conductivity meter. Again, I'm showing one thermoscientific conductivity meter. This conductivity meter is also made by different international and national companies in India. Okay, now the next important thing is soil organic carbon. Now, the soil organic carbon is very important part. If the soil organic carbon in your soil is high, that means your soil is highly fertile. And if the soil organic carbon is low, that means your soil is non-fertile. So soil organic matter measurement or soil organic carbon measurement solely can give the indication of fertility status of your soil. Okay, and what is the method we use for measurement of soil organic carbon? Generally, we use the Walkley and Black method, which is a titration method to measure the soil organic carbon. Okay, so what is the principle of measurement of soil organic carbon using Walkley Black method? So what happens? We know that soil contains some amount of organic carbon. So to measure that organic carbon, we need to convert that carbon to carbon dioxide. So how to convert this carbon to carbon dioxide? We need to oxidize the carbon. So to oxidize the carbon in the soil, we need some kind of oxidation agent or oxidating agent. So what are these oxidating agents? Oxidating agents are potassium dichromate as well as concentrated sulfuric acid. It is a mixture. Potassium dichromate, it is a dark red color chemical, and also concentrated H2SO4. So this mixture acts as an oxidizing agent, converts this carbon into carbon dioxide, and the excess of K2Cr2O7 or potassium dichromate, which is not reduced by SOC or which was actually not used for oxidation of carbon, was was back titrated against the standard ferrous ammonium sulfate in the presence of some indicator called diphenylamine indicator. So we basically take this potassium dichromate in excess amount. Some amount of this potassium dichromate converts this carbon to carbon dioxide, and rest of the potassium dichromate left, unreacted. So we calculate this unreacted fraction of potassium dichromate by titrating it with ferrous ammonium sulfate in the presence of an diphenylamine indicator. Okay, and these are the so once we calculate the unreacted fraction of potassium dichromate, then we can obviously understand what is the amount of potassium dichromate which has actually reacted, and from there we can calculate the carbon concentration in the soil. And these are the reactions which are involved. I'm not going to their details. Okay, you can later take a look what are the reactions which converts this carbon into carbon dioxide by mixing with potassium dichromate and H2SO4. And these are the steps. For example, we need to take first 1 gram of soil, then we are adding 10 ml of 1 normal potassium dichromate and 20 ml of concentrated sulfuric acid, and then the beaker is swelled and allowed to stand for 30 minutes, and then we add 200 ml of water and 10 ml of concentrated phosphoric acid, and then we add these diphenylamine indicator 10 to 15 drops, and then titrate against the freshly prepared ferrous ammonium sulfate. Why freshly prepared? Because if we keep them too long, it will convert to ferric from ferrous. So we freshly prepared ferrous ammonium sulfate and then titrate this solution and until the color changes from violet blue to green. And simultaneously we do a blank titration also. And the calculations are also given here. Now you understand one thing, using this Walkley-Black method, we can get only 77% of the soil organic carbon. So the recovery ability of this method is only 77%. So how to get this 100%? So we have to convert, we have to multiply it with 1.3 to get the 100% result or corrected results. Okay. And from there, we also can calculate the organic matter content in the soil. Okay. Now, what is sufficient, efficient, or low? Okay. So, based on the soil organic carbon interpretation, if your soil is contain less than 0.5% of organic carbon, then your soil is considered as a low in organic carbon content. If it is containing 0.5 to 0.75, then it contains medium content. And if it is greater than 0.75, then we call it as a high content of nutrient. Okay. So this is how we interpret the soil organic carbon results. Okay. The next important is available nitrogen. Available nitrogen we generally measure by Subaiya and Asija method, who gave this method in 1956. Now, nitrogen availability to plant depends on its mineralization, and I have already told you that the plant available nitrogen is basically this ammonium and nitrate, because these are the only two forms which plant can extract from the soil. Okay, so basically if we measure this ammonium and nitrate in the soil, we can measure the available nitrogen. So what is the principle? So a known weight of soil is treated with an excess of alkaline potassium permanganate, which extract easily oxidizable fraction of organic matter. Ammonia evolved is absorbed in a known volume of standard acid, and then excess of which is titrated against standard alkali using methyl red as an indicator. Okay, so again the reactions are given here. I'm not going to discuss them in details. Okay, so you can always take a look later on, and step by step all the reactions are given here, and also the steps of all the all these steps are also mentioned here. How step by step how much soil you should take, and then what amount of this potassium permanganate you should apply, and all these different types of steps, all these different important steps are mentioned here. Okay, so if you can take a look later on. And these are the calculation, how to calculate this available nitrogen. Remember, this based on your results. So what are the low, medium and high content? So after the calculation, if you are getting something which is less than 272 kg of nitrogen per hectare, then your soil is low in nitrogen. If it is greater than 544, then your soil is high in nitrogen. And if your soil is in between 272 to 544, then it is considered medium. Now, obviously, remember that a well-balanced fertilization of your soil should always focus on this medium. You should not, your soil should not have low nitrogen content. Your soil should not have very high nitrogen content also. Because if your soil has very high nitrogen content, that will create nitrogen toxicity in the plant. So what will happen? When there is a nitrogen toxicity, the plant will lodge, okay, and there will be huge crop loss, okay. So always you should maintain the medium level of fertility. So if you after the analysis of your results, if you see that your soil is contain low amount of nitrogen, you ask your farmers to apply more nitrogenous fertilizer. And if your soil already have high amount of nitrogen, you ask your farmers not to apply nitrogenous fertilizer for at least 1 or 2 seasons, so that this high amount of nitrogen can be utilized by the crops. And if it is contain medium amount of nitrogen, you ask your farmers to do nothing. So he doesn't need to do anything. When the medium is there, so it is already optimum. Okay. Now we do this available nitrogen measurement in the lab using this automated Zeldal method, and this is an automated Zeldal distillation unit which you use in the lab for measurement of available nitrogen. Okay, we can program it and it will automatically calculate and give you the results of available nitrogen content. Okay, the next important thing is available phosphorus. Now remember that phosphorus is one of the major plant nutrient which plants uptake as primary and secondary orthophosphate. So the primary orthophosphate is H2PO4 ion and secondary orthophosphate is HPO4 2- ion. Just like in case of nitrogen, phosphorus also is available in these only 2 forms, H2PO4- and HPO4 2-. Okay. Now this H2PO4 2- is dominate in acidic range of soil and HPO4 2- dominate in alkaline range of the soil. Now remember that at pH 7.2, that means near the neutral pH, these 2 ions have equal activity. So that is why I said earlier that you should maintain a near neutral condition as far as the pH of the soil is concerned. Okay. Now depending on the, so you understand why we measure the pH, because unless you have an idea about your soil pH, you cannot go for the further steps. So you can see Bray and Kurt's method is used for acidic or to neutral soil, and in case of alkaline soil we use the Olsen's method. So unless you know the pH of your soil, you cannot go with this method. So that is why at the beginning of the soil testing program, we measure the pH of the soil. That is why it is called master property or master determinant of the soil. Now you see these are the Bray and Kurt's method. The principle and reagents are already given here. Again, I'm not going to discuss all these detailed steps here. You can always go back and see what is the principle. So remember, Bray and Kurt's number 1 method is generally used for acidic soil. So if we are measuring the phosphorus content in the Kharagpur area, which is acidic soil, then we'll always go with the Bray and Kurt's method number 1. But if we are going for some soil samples from Punjab area where the soil is alkaline, then we'll go with the Olsen's method. Okay, so here the Bray and Kurt's method, and there all these important steps and calculations are already been given. So I'm not going through in details about this, but remember whenever we do different types of, when we execute different steps in this available phosphorus estimation, ultimately there is a color development. So if your soil is containing high amount of phosphorus, you will get a high concentration of a color. And if your soil is containing low amount of available phosphorus, then the color development will be somewhat low. So depending on the phosphorus concentration, the color intensity of the extraction or extracted solution will change. Okay, so based on that, so once we extract those colored solution, then we measure the intensity of those phosphorus in those color solution using some meter. We call them spectrophotometer or colorimeter. Okay. So the calculation for Bray and Kurtz method is given here. Okay. So based on your calculation, if you are getting something which is less than 20, if your amount of, okay, one thing you must understand, whenever we consider phosphorus measurement or recommendation in the soil, we generally measure in terms of P2O5 or phosphorus pentoxide. And it has been a norm for using this phosphorus pentoxide since the British period, because this is used for measurement of nutrient content in the fertilizer. So that is why we express the phosphorus measurement of phosphorus test in terms of phosphorus pentoxide or P2O5 per kg of hectare. So ultimately you will see that available phosphorus, once you calculate the available phosphorus, you can convert into available P2O5 also per kg per hectare. All these calculations are given in the slides. Okay. So once you calculate and you get that available P2O5 is less than 22.5, then your soil is deficient in phosphorus. If it is greater than 56, then it is high in phosphorus. And if it is somewhat in between 22.5 to 56, then it is considered as the medium concentration of phosphorus. Okay. Now let us see what is colorimetry and spectrophotometry. Okay. Now once we extract this phosphorus using some extracting solution, in our case either Bray and Kurtz number 1 or Olsen's reagent, we will see some color solution, and then we measure this color intensity using an instrument called spectrophotometer. Now we want to understand what is spectrophotometer and how it operates. Okay. Now there are two terms. One is called colorimetry, another is called spectrophotometry. Now colorimetry is the use of human eye to determine the concentration of colored species. Okay. So, and spectrophotometry is the use of instruments to make the same measurement, and it extends the range of possible measurement beyond those that can be determined by the eye alone. So our eye only can see what is visible. Okay. Now, if you see the total electromagnetic radiation or electromagnetic spectrum which is available in the universe, we divide them into several regions, starting from gamma rays, X-rays, UV rays, visible light, infrared light, and radio waves. Okay. Each of them has their own specific interval of wavelength. For example, in case of gamma ray, it is having very short wavelength. X-ray is having longer wavelength than gamma ray, but shorter wavelength than UV ray. UV ray has longer wavelength than X-ray, but shorter wavelength than visible light. And visible light has wavelength from 400 to 700 nanometer. Okay, and our eye can only see in between this 400 to 700 nanometer because all the colors are basically perceptible from this visible wavelength range. After the visible wavelength range, this starts infrared, which is also not visible to in our eye. UV we cannot see, X-ray we cannot see, gamma rays we cannot see. Also, after the infrared, there are radio waves which also we cannot see. So colorimetry is basically a form of spectrophotometry. So in case of spectrophotometry, it can measure the intensity of the color absorption or intensity of the color in any of this range, from starting from UV to visible to infrared. So any of this wide range, for each individual wavelength, it can measure the color intensity. But for colorimetry, since the name indicates colorimetry, it can only be useful within this 400 to 700 nanometer range. So colorimetry is a shallow, narrow term which is only useful within 400 to 700 nanometer. But spectrophotometer can be used from ultraviolet to infrared. Okay. Now, this is an example of a colorimeter made by Systronics, which is an Indian company. And this colorimeter, so what happens in this colorimeter, you pass a light through this solution, and obviously depending on the material concentration in this tube, we call it cuvette, it will absorb some amount of light and the rest of the light will be passed through. So this colorimetry will measure how much amount of light is getting absorbed by this colored solution. See, one thing is very clear. This is a colored solution, which we can see. So that is why it is called colorimeter. But spectrophotometer, which is a broader term than colorimeter, can measure both colored as well as uncolored solid solution also. Okay. So spectrophotometer is an instrument that measures the amount of light absorbed or the intensity of the color at a given wavelength. And the intensity of the color can be given a numerical value by comparing the amount of light prior to passing it through the sample and after passing through the sample, and this quantitative measurement of light absorbed are the transmittance and the absorbance. So spectrophotometry says that, so let us consider this is a colored solution. In this colored solution, this is a yellow colored solution. Okay, so the light is entering here and the light intensity is I0, and the light which is passing through, it is intensity is only I. So the path length of this total solution is only B. So, and here path length means the length that light has to travel. So here you can see the path length is B. So, and the absorbing, the sample concentration is C. So here you can see the transmittance is given by the equation T equal to I by I0, where I is the intensity of the light after it has gone through the sample and I0 is the initial light intensity. And absorbance is the related to percentage of transmission. So you can calculate the absorbance by taking a negative log of transmittance. Okay, just like here. So why we use this spectrophotometry or why we use this spectrophotometry for measurement of concentration? Because it is based on the Beer's-Lambert's law, also known as Beer's law, known as the linear relationship between the absorbance and concentration of an absorbing species. If A is the absorbance, it is basically the factor of three important components. A, which is a molar absorptivity, B, which is a path length, I already showed you path length in centimeter, and C is the concentration. So if the path length is constant and if the molar absorptivity is constant, that means absorbance will be proportional to the concentration. That means if the concentration is increasing, that means their absorbance will also increase. So that means if the color development is increased, that means the solution will absorb more and more color. Just like here you can see, this is called a cuvette, and we put a solution here and we pass a light, and based on the amount of light absorbed, we can say that this is the concentration of that particular element in the solution. Okay, so basically when we do this type of colorimetric or spectrophotometric measurement, we develop a standard curve. By, for example, we make a solution of 0 ppm, for example, 0 ppm phosphorus, 2 ppm phosphorus, 4 ppm phosphorus, 6 ppm phosphorus, 8 ppm phosphorus, and then we measure their absorbance. These red dots are those absorbance, and then we fit a straight line. So this is called a standard curve. Okay. So this standard curve is giving you the, this standard curve you need to build before you measure any unknown sample. So once you develop this standard curve, any unknown sample, suppose you are collecting an unknown sample and you are measuring their absorbance. So suppose their absorbance is 0.3. So you put it here and it will give you corresponding concentration from this x-axis. So this is how we measure the concentration of a particular nutrient based on the standard curve and spectrophotometry. So this is called a spectrophotometer. This is a simple spectrophotometer. This is an instrument that measures the amount of light that passes through the sample, and they have different parts like display, they have wavelength selection, they have sample holder. We also have knobs or buttons used to calibrate the specs to measure the designated molecules, so we can control all these things. And this is a simple schematic of spectrophotometer where there is a lamp, and after that there is a lens. After the lens, so the lamp will emit the colors of light. So this is basically light source, and monochromator is basically selecting one wavelength. So the light can contain different wavelengths, and monochromator can only. So monochromator is basically a filter. So this filter will filter only one wavelength, and this wavelength will pass through this color solution and ultimately get detected by this detector, and which will be further amplified and ultimately we'll get the results. So this is how we do this. This is the working principle of spectrophotometer. Okay, so this spectrophotometer is used for measurement of sulfur, measurement of phosphorus. Also, it can be used for measurement of nitrogen by some methods. Okay, there are some methods we also use spectrophotometer for measurement of available nitrogen, like calcium chloride method. Okay, so different types of spectrophotometers are there in the market, like ultraviolet spectrophotometer are there, visible spectrophotometer are there, UV visible spectrophotometer are there. Okay, so depending on the range on which you want to work, it will, these different types of spectrophotometers we generally use. Okay, so this is an example of UVV spectrophotometer which you use in the laboratory, which is a sophisticated instrument. Okay, it is also costly than simple spectrophotometer. Okay, the spectrophotometric schematic I have also already told you. Now let us go to the next important thing, that is available potassium. Now potassium is also abundant in nature. However, plant available potassium is very much limited, and generally we extract the potassium using the neutral normal ammonium acetate solution, and then we measure their concentration in the instrument called flame photometer. Okay, so this instrument is also very standard instrument for any laboratory, soil testing laboratory. Okay, so this principle and steps are given here. I'm not going to discuss them in details, but remember that available potassium, their concentration based on the, just like in case of phosphorus we express their concentration in terms of P2O5 per kg per hectare. Similarly, in case of potassium, we represent their concentration in terms of potassium oxide or K2O kg per hectare. So if your results come below 136, that means your soil is low in potassium, you should apply potassium. If it is greater than 337, then it is high in potassium. If it is somewhere between 136 to 337, then it is medium in potassium. Remember, you need to apply potassium when it is low, and you do not need to do anything when it is medium or high. Okay. Now the instruments which you use for measurement of potassium is called the flame photometer. Okay, so flame photometer also can measure sodium, potassium, lithium and calcium, and we call it flame photometer because here the source of energy is a flame which is generated by liquid petroleum gas. Okay, so it is a control flame test with the intensity of the flame color quantified by photoelectric circuitry. So basically, once we extract the potassium in the solution, we aspirate it, we make an aerosol in the flame photometer, we vaporize. So when this aerosol goes in contact with the flame, it will vaporize and it will convert into molecules. And when more energy from this flame is coming, it will dissociate into the flame by taking the thermal energy. And ultimately what will happen, you know, there are electrons in the ground state. These electrons will be energized and they will go to higher excited state, and after a certain period of time they will further go back to their original state. And when there will be transition from this upper state to lower state, they will release some energy, and that energy will be measured by this flame photometer. And the intensity of this emitted energy is based on the concentration of a particular element. Okay, so the sample is introduced to the flame at a constant rate. Filter select which colors of the photometer detects and exclude the influence of other ions, and flame photometer works on the emission spectrophotometry principle. Okay, so you will see that flame photometer. So flame photometer for potassium, it will be on the emitting wavelength is 766, and it will generate a violet flame. If you are selecting 670 filter, it will be useful for measurement of lithium, which will give a red flame. If you are selecting 622, you can measure calcium, it will give a orange flame. In case of sodium 589, it will give a yellow flame. In case of barium, it will be 554, it will give a lime green. So basically, based on the emitted wavelength range, it will give you a specific color, and the color intensity will give you the idea about the concentration of that particular nutrient. So this is the internal construction. First, we nebulize the sample. So this is a sample. There is an aspirator. So the aspirator will suck the sample here, and there is a nebulizer. So this nebulizer will inject the sample in this mixing chamber. In this mixing chamber, there will be air input. So this air will mix with this sample to make an aerosol. This aerosol will go to this burner where there is a flame is generated, and after it gets the energy from this flame, it will emit a light and flame, which will further go through this lens and further filter and photodetector and amplifier, and finally with the readout. Okay, so this is how this instrument basically works. Okay, available sulfur. These are the steps of measurement of available sulfur. You first weigh 5 grams of soil in then 16 ml of shaking bottles and 25 ml of 0.15% calcium chloride solution. Shake for 30 minutes, and then step-by-step methods are already given here. Okay, so we have seen available nitrogen, available phosphorus, available potassium, available calcium, available then available sulfur, and then next comes the micronutrients. So the micronutrients here, I will just concentrate on these 4 major micronutrients: iron, copper, zinc, and manganese. So these 4 major micronutrients is basically extracted by an extractant called DTPA. The full name of DTPA is diethylene triamine pentacetic acid. And once we extract these available forms of iron, copper, zinc, and manganese, we measure their concentration using an instrument called atomic absorption spectrophotometer. Okay, so this atomic absorption spectrophotometer is another sophisticated instrument and costly instrument also, which you can use for measurement of all these different elements and also some heavy metals. So what happens in this in the short form, we call it AA or AAS. So atomic absorption is based on the principle that a ground state of atom is capable of absorbing light of the same characteristic wavelength, and it would emit if excited to a higher energy level. So what happens in the ground state? From the ground state, if any electron is absorbing some light, it will absorb and it will go to the higher state. And this absorption of light is very, very fixed for a specific element. From there, and based on the intensity of the absorption, we can say how much quantity of that particular element is present in the sample. So this is the principle of atomic absorption spectrometer, and this spectrometer is widely used in different soil testing laboratories. Remember, this is very sensitive for most of the elements, and concentration up to ppm level you can routinely determine using the frame atomic absorption system. That is the basic one. You can attach another furnace called graphite furnace, which generally gives higher temperature. So when higher temperature is there, it will be more efficient, and then you can get the results up to parts per billion. So this is how this instrument basically works. And this is the interschematic diagram of atomic absorption spectrophotometer. It first goes for sample uptake, then nebulizes it, it goes to cloud chamber, and then goes to burner. After it goes to the burner, there is a source light coming from hollow cathode lamp. Ultimately, it goes to monochromator or filter, and then ultimately to detector, and then amplifier, and finally we'll get the output in PC or printer. Okay, so this is how this instrument basically works. Now, what is the radiation source? The radiation source of this instrument is called the hollow cathode lamp, or HCL. So this is used because the requirement of a source of narrow lines of the correct frequency. These individual element has their own hollow cathode lamp, and this hollow cathode lamp will basically generate the specific wavelength of energy or light which will be absorbed by the particular nutrient. So for each of the element which you want to measure, we have to use their specific hollow cathode lamp. Okay, so these are other details of hollow cathode lamp. Another advanced technology which generally people are using for long time, it is called ICP AES. Now, ICP AES is the short name of inductively coupled plasma atomic emission spectrometry. It is a highly sophisticated method, and it is based on the emission spectrophotometric technique. It is more precise and accurate than atomic absorption spectrophotometer. So ICP is more accurate than AES, but it is more costly. Okay, why it is more accurate? Because it can measure, the principle of ICP generates huge amount of temperature up to 6,000 Kelvin, which is a huge amount of temperature in which all the elements can be estimated without any interference. So it is more precise, and using ICP you can go up to parts per billion level also. There are the principle of ICP I have given here. I do not have enough time to discuss this principle right now. So please go through this principle, in details. All these are given in the slides. Okay. So this is an ICP instrument, which, it is also very, very costly, 40 to 50 lakhs of rupees, one instrument cost. So very sophisticated research labs, you can see this type of instrument. Okay. Atomic absorption spectrophotometer is way cheaper than ICP. Okay. So I'm not going to details about this ICP. Now, one thing I should mention, that you see that I have showed you different extractants for extracting different nutrients, right? For phosphorus, for potassium, for micronutrients, for sulfur. Now, there is nowadays one method called MELIC3 method. It is basically a combination of different chemicals like ammonium nitrate, ammonium fluoride, EDTA, which is a chelating agents. Full name is ethylene diamine tetraacetic acid, then acetic acid, nitric acid. Okay, so all these are combined together to form this extracting reagent. This is called MELIC3 extracting solution, and we can use them together to extract phosphorus, potassium, calcium, magnesium, and also micronutrients. So this is how one go. I mean, we do not need to separately extract all these. If we use this MELIC3 extracting reagent, we can extract all these phosphorus, potassium, calcium, magnesium, and micronutrients except for nitrogen, we can measure all the other nutrients in a single extraction. And then we can measure using either ICP or using the AAS. Okay. So this is how about the standard methodologies of soil testing, which we generally follow in the standard soil testing lab. Another thing is there that is called soil testing kit. Now, soil testing kit is basically a box which is portable. So it is not as accurate as it is not accurate. It is a qualitative estimation of soil, fertility status. So it is a portable soil testing kits they enable farmers to take it field and take the measurement. So if a farmer want to take the measurement of their soil directly on site, he or she can use this soil testing kit. Okay, so these soil testing kits, most common kits helps to find the pH and primary nutrients like N, P, and K. And kits can also help in finding all nutrient, but is expensive. See, these kits are basically what contains. They basically contain several small bottles, and they will have an reagent. So all these different reagents are there. There will be a instruction of how to measure different elements. So there will be droppers, there will be basically test tubes. So there will be scoop to take soil samples. So they will instruct you step by step for each of the parameter. First you take 1 scoop of soil, then you add 5 ml from reagent number 5, then you add 4 ml from reagent number 2, then wait for 5 minutes. Then you see the color. So once the color is developed, it will also have a color chart. So you have to see the best color matching between this developed color in the solution and the color chart, and from there you can get an idea about whether it is deficient, sufficient, or medium in quantity. So these reagents are meant for certain numbers of soil samples. For example, you can measure up to 50 or 100 number of soil samples. After these reagents are exhausted, then you have to go for then you have to procure more new these reagents which are expensive. So soil sample is so this is only qualitative. For more reliable measurement, you should send your soil samples directly to the lab. Okay, now we have seen what are the steps of the soil testing. Now the important you can see this how the soil test results looks like. So you see the soil sample details, date of sample collection, survey, and all these details: farm size, irrigation status, their position, latitude, longitude, all these given, and then their individual parameters which they have tested and whether they are moderate, normal, or low, very low, deficient. All these are given here, and their cutoff normal values are also given. Just like in case of any testing, our diagnostic testing of human being, also will be getting the normal level, and also whether the interpretation whether our soil is moderate or low or very low or sufficient or deficient, all these type of things. Okay, see here we are getting 1.65 ppm of copper, and normal level is greater than 0.2 ppm, so we have sufficient. Similarly, for available manganese, greater than 2 ppm is sufficient. So available manganese, it is we are getting 6.41. So it is sufficient. However, in case of iron, the sufficient level is greater than 4.5 ppm. However, our soil contains only 0.71 ppm, so it is deficient. So this is how we interpret our results. Once we do the interpretation, the next important thing is fertilizer recommendation. Okay, now you see here, this is a fertilizer recommendation chart for different crops. Let us consider rice. Okay, here you can see Kharif medium duration 125 to 135 days. High yielding varieties Jaya, Ajay, Kunti, Vikramacharya, Prohash, Pratap, IR20, all these different varieties can have this recommendation. So based on this fertilizer recommendation, so once you have all these soil test results, you are getting whether your soil is having high level or high, low, or medium level. Okay, so you can see in this chart also nitrogen P2O5 and K2 are given, whether it is high, medium, or low. Okay, so based on this chart, you can identify how much you should apply, fertilizer you should apply. So you see whether, suppose in case of nitrogen, your fertilizer, your test results came high. So high means for you should apply 50 kg of nitrogen only. Okay, you do not need to apply more than that. If it is more than that, there will be loss. If it is medium, you should apply only 60 kg of nitrogen. So similarly for each of them. So from this chart, you can calculate how much fertilizer you need and how to apply this fertilizer. For example, for nitrogen, you apply 1/4 of nitrogen as basal, full potassium and phosphorus, potassium as top dressing for nitrogen, and then half nitrogen you apply at 15 days after transplanting, and last 1/4 you apply at 40 to 45 days after transplanting. So this is how you do the recommendation, fertilizer recommendation. If you have this chart and if you have this fertilizer recommendation in your hand, then you can calculate how much fertilizer you need to apply. So you see one thing, that why soil testing is important. Soil testing is important because if your soil is already having a particular nutrient in sufficient quantity, you can save a huge amount of money by not giving fertilizer. So you can save a huge amount of money. Also, if your soil is having deficiency in particular nutrient, you can apply those nutrients to get more yield. So these type of things are important. So that is why soil testing is very, very important aspect. So I have given all these different types of crops here. You can take a look. Fertilizer recommendation for cauliflower, not only for macronutrient, here micronutrient recommendations are also given here. So this is a fertilizer recommendation for the soil health card, which is one of the which has been started by Department of Agriculture a couple of years ago. Okay, so this is how we do the traditional soil testing. Now I am going to briefly tell you about how to set up a soil testing lab. Okay, so if you want to set up a soil testing lab where you want to measure NPK, primary nutrients, secondary nutrient, micronutrients, pHs, electrical conductivity, organic carbon, then the laboratory should have the capacity of testing around 350 samples per day. And this way around you can analyze 1 lakh samples per year. And the target for the testing of the NPK is only 80% of the total sample. And for micronutrient, we should target up to 20% of the total sample. However, the capacity of utilization can be considered as 40, 60, 75 and 80% in the first 4 years, and 5th year onwards it will continue to be 80%. So first year you should not, you will not utilize the 100% capacity. You can utilize only 40% of its capacity. The next year 60%, then 75, third year 75%, fourth year 80%, and then it can go up. So you have to calculate your cost involvement based on that. So what are the equipments you need to set up a laboratory? You need an AS, one Zeldal unit for nitrogen distillation, one spectrophotometer, one flame photometer, one conductivity meter, one pH meter, one nephelometer for sulfur estimation. We can measure the sulfur by turbidimetric method also. So for that method, we can also use nephelometer. Otherwise, you can measure using spectrophotometer also. Rotary shaker for shaking the sample solution. You need electronic balance, you need analytical balance or top loading balance. You need drying ovens to dry the glasswares. You need computers or fax with appropriate softwares and printer to generate the recommendation curve. You need tabletop centrifuge, you need gas telephone connection, glasswares and chemicals also. So these are the different requirements. This is the shaker, this is a laboratory balance which you require. Transport, as the laboratory is a static, it is a one single place, there is a need for collecting and transporting the soil samples to the facility, and as the awareness about the facility gradually builds up in the nearby areas, the farmers would come to the laboratory with the soil sample by themselves for testing, and the trainees or vocational courses may also be deputed and their service may be utilized for collection of representative soil sample as they transfer to them the nearest laboratory. Raw material, what raw material? Chemicals and glasswares are the raw materials, and these are these are, you can get these chemicals and glasswares from any, standard scientific equipment manufacturers or suppliers. Okay. Manpower needed. Generally, for a large, scale soil testing, you require one postgraduate in agriculture with soil science specialization, or with an adequate experience in soil science, and be the lead analyst for the laboratory. And also, he will be supported by the 2 lab assistant from soil science or agriculture background for preparation of soil samples and other laboratory works. And for collection of samples and field related works, 3 persons are also required. Techno-commercial parameters. So a team of 1 specialist and 2 semi-skilled workers can analyze up to 350 samples per day. So if we consider we are doing 6, analysis are being done for 6 days in a week. So sample collection, a report writing, etc. will be done simultaneously. So monthly, approximately 8,750 samples will be analyzed, and yearly 1 lakh samples will be analyzed. So sample collection will be done only 8 months in a year, depending upon the cropping pattern. Analysis can be carried out throughout the year. I have already told about you the capacity and charge. What will be the charge of testing? The charge of testing will be rupees 15 per sample for analysis of NPK, and 850 per sample with micronutrients. And what is the chemical and glassware consumption? For macronutrient, we require 7 rupees per sample, and for micronutrient, we require 100 rupees per sample. And we are charging 850 rupees for sample with micronutrients, and only for macronutrients rupees 15. So this is a standard layout of a soil testing laboratory. As you can see, there is an analysis room 26 by 24, and there are some instrument room, because you need to keep the instrument into a safe room 22 feet by 12 feet, and there should be a chemical store. There should be office where you should collect the soil sample. There should be soil store. So all these informations are given in this layout. Okay. Next important thing is financial involvement for setting up a STL. So for the whole project, cost comprises of around 53.65 lakhs towards capital cost, and rupees 6.26 lakh for operational cost in the first year. And the operational cost in the second, third, fourth year onwards is 11 lakhs, 12 lakhs, and 12.61 lakhs. So you can see plant and machinery, it cost around 35.28, miscellaneous fixed asset 14.70, total fixed asset around 50 lakhs, working capital 6.26, and then pre-operative expenses 3.69. So total, the expenditure will be 59.91. Okay, financial involvement. Then you can see testing of 1 sample, we are charging 15 per soil samples and 850 per soil sample with micronutrients. So how much revenue we can generate? So tentatively, I have calculated that the revenue in the first 3 years and 4th year onwards would be 41 lakhs, 62 lakhs, 77 lakh, and 82 lakhs respectively. And this is the cash flow statements, where you can see what is the capacity of utilization when it is increasing, and then soil test, how much, you are doing the NPK testing, micronutrient testing, how much you are generating, how much expenses you are doing, and what is the net income. I have already told you these values. Capital cost are equipment 28.6 lakh, chemicals, glassware 4 lakhs, contingency 2 lakhs, standby generator or electricity source is 4 lakhs, and total is 38.6 lakhs. And for this, you can get a subsidy about the 50% of the project cost, limited to maximum 15 lakhs as one-time subsidy. Cost of equipments I have given here is contains around 16 lakhs. So total 20x 8.6 lakhs will be required for procuring different equipments. And some indirect costs are there, like power, water, manpower, technician cost, their salary, chemical, glassware, LPG, repair, maintenance, telephones, travel and canvassing, collection of samples, annual collections. So all these are given here. Okay. So all these informations I have given in this slide, so you can take a look. And these are some references which I use for preparing this lecture. If you are interested, you can always go back and see this more information. Obviously, within these 2 hours, it is not possible to discuss all of these in details. And if you have any question, you can email me. This is my email, somzcall@gmail.com, and my telephone number is given here. So if you are interested to do this kind of business venture, you are most welcome. And so this is about my presentation about the soil testing, and this is only about the traditional soil testing. Okay, so if you have any question, you can ask me. Okay, so I can see that some questions are there. In Tamil Nadu, the lab report also reflects NPK, iron, manganese, zinc, copper, and boron you showed in the color chart and not other nutrients. Any reason for selecting only these nutrients? In Tamil Nadu, lab report also reflects NPK, iron, manganese, zinc, copper, and boron in the color chart. See the lab report. See the NPK. If you can measure the nitrogen, phosphorus, potassium, iron, manganese, zinc, and copper, and boron, you are basically covering the most of the nutrients which basically which basically are important for the crop growth. So if you are having this information, most of the time you will have an idea about your soil fertility status. Okay. Chlorine, it is not that important to measure. Molybdenum, most of the time it is present, but most of the time you will see the deficiency coming from iron, manganese, zinc, and copper, and boron. Okay. So if you have any other question, please inform. Okay. I do not know, Professor Machavaram, whether we'll send this presentation to the participants or not, whether there is a provision or not.
Yeah, it is already in YouTube Live. They can already given the answer that they can just go through that.
Okay. So thank you. I hope that this information is helpful for you. If you have further information, I'm always there to help you. Please email me, and I'll be more than happy to reply you with more information. Unfortunately, I am not able to show you the advanced methods which we are nowadays using for cost-effective soil testing. But I'm sure that when in future, when you will be able to visit our campus in physically, I'll be giving you a full tour and give you how we are nowadays using mobile phones to capture the image of the soil then and there in the field, and based on this image we can measure different types of soil properties. I can show you that. Okay, if we have a time, if we have the chance later on. Okay, so thank you, Professor Machavaram, over to you.
Thank you.
Yeah, thank you, Professor Som, for your excellent lecture on the soil testing related to the crop production. Now I request the participants, if they have any questions or queries, they can interact with you.
Mm-hmm. Yeah, if you have any question, please ask.
Yeah, from the participants.
Yeah, sir, one question. So this is about the micronutrients in the soil. Is there a way to visualize the micronutrients that live in the soil in a graphical representation? Right.
Well,
I want to look at the dynamics of yeah.
See, micronutrients cannot be directly visualized. You cannot visualize any of these nutrient sufficiency or deficiency just by seeing the soil itself. Okay, so they are visible. Their deficiency sufficiency symptoms generally are visible in the plant part. Okay, so if the soil is deficient in that particular nutrient, plant which are grown on that area will give you this. Will give you the indication. So instead of looking directly into the soil, we can develop some technology where we can use some apps in our mobile phone to take the image of the plant, and by analyzing their deficiency symptom, we can definitely say, okay, we are seeing these deficiency symptoms. That means this soil may be deficient in boron. This soil may be deficient in iron. So we have already developed in our laboratory. We have already developed this kind of application, Android application, in which you can take your mobile phone, you can take an image, and by analyzing the image of the plant, you can have an idea about the nutrient deficiency. But as such, by looking into the soil, it is not possible because these micronutrients are very, very small. Okay, and if you have also, I have seen some materials. I have seen some materials where they have developed some instant color development extracting solution. So once you develop that instant extracting color solution, you can use your mobile phone as a spectrophotometer, and then you can measure the intensity or concentration of the nutrient then and there in the field also. But all these are in the in the research phase. So far in India, only IIT now doing this advanced research. Professor Machavaram is also doing these optical sensing methods for different agricultural applications, and I'm doing here for soil application, soil nutrient measurement. Hopefully, within a couple of years, we'll be able to generate a foolproof system for utilizing these Android app for image-based analysis of soil parameters. Okay.
Okay, thank you, sir. And you talked about the applications, right? Mobile application. Is that something downloadable or we can have access to?
No. It is in the development phase, and my students are doing that. We have already proved the utility of this mobile app for soil organic carbon and texture. And nowadays we are doing the research for macro and micronutrients. Once we are satisfied with our results, then we'll be we'll see that app in the platform. Okay, the Android platform.
Okay, thank you so much. Thank you so much.
Sir, good evening. This is Motesh Mohan from Nilgiris, Tamil Nadu, sir.
Okay.
Sir, what is the correct CN ratio for vegetable crops in acidic soil, sir?
CN ratio you are talking about?
Carbon nitrogen ratio, sir.
Say carbon nitrogen ratio generally varies from, generally the standard ratio is 20:1. So you should maintain something like that. 20:1 should be maintained, because if the carbon nitrogen ratio is greater than 20:1, then there will be problem of immobilization. So generally, for a good supply of nitrogen throughout the year, we generally recommend that you should maintain the carbon nitrogen ratio of 20:1, and for that you should apply some green manure, some compost, which will help you to get that optimum carbon nitrogen ratio. Remember, less carbon nitrogen ratio is always better, but higher carbon nitrogen ratio is problematic. Okay.
Thank you, sir.
Any other questions?
Anyone who is interested to interact?
Hello.
Yeah.
Yes.
Yeah, hi, sir. This is Praful from Anantapur district. We are in a dryland agriculture. We are trying to grow vegetable crops through organic systems here. And we use only compost for growing vegetables, and we don't use any amendments. So we are trying to grow cruciferous vegetables like cauliflower, cabbage, and we are seeing a deficiency in them because they are not able to form heads. So is there any organic way to supplement the soil for that?
See, sulfur containing concentrated organic manure are there. Okay. For example, some groundnut cakes are there, some currant cakes are there. This concentrated oil cakes you should apply, because these contain sulfur, and when you grow this cruciferous crop, they require sulfur for their growth. Okay. Correct. Because the oil content is dependent on the sulfur, okay, in the soil. So you should more focus on some new organic source which contains more amount of sulfur. And these groundnut cakes and these different types of cakes or concentrated organic manure are very much helpful for supplying that. Make a literature search, and you will be having a good idea about what are the available these cakes or concentrated organic manure in your locality, and apply those. Okay, instead you are applying compost, that is fine, but you should apply these oil cakes also, because these oil cakes contains huge amount of sulfur, which will help in the development of the head. Okay.
Thank you so much, sir. Thank you.
Okay, I think they don't have any further question. Okay.
Okay, thank you, Professor Song, for an excellent lecture once again, and I thank all the participants who have participated online via Google Meet as well as a YouTube Live. If you have any query, you can directly post it, or you can even interact with us over the sheet, whatever we have shared. Thank you once again. Tomorrow we have another session at 4 p.m. by Professor D.K. Swing sir. It is on organic farming and vermicomposting. You can join again tomorrow. Thank you all.
Okay, thank you.
Thank you, Professor. Yeah.
Sir. Hello.
Yes, Sukant Das?
Yes, sir. Sir. Hello.
Yes, please.
Sir, actually, yesterday I did not get attend the meeting. Actually, I have not get that Google Meet option. So I am interested, sir, actually in biofuel and biogas. Also
No, actually, that is on 15th. You can attend for that on 15th. Actually, we have just rescheduled that one on 15th. Yesterday, it was on micro irrigation. Okay.
Sir, actually, biofuel and biogas. Sir.
Yeah, it is on 14th of December.
14th,
We can attend that. Not 14th, 15th. Okay.
15th, sir, no?
Yeah. Even if you miss that also, the lectures are there online, YouTube. You can go through that. Okay. Okay. Thank you. Yeah, you can leave. Thank you.
Hello, sir.
Yes.
Sir, I just want to know, I haven't attended online class yesterday.
Yeah, it is there.
I see that in later in YouTube channel.
Yeah.
So I want to know if there is any feedback form for this also.
The feedback form already we have circulated yesterday for yesterday's session. You can fill that for today. It is not an issue.
I filled for today, but
yeah, that is sufficient. No problem. Yesterday you were not there. Not a problem for that.
Yeah.
Okay.
Okay.
We have indicated 90% attendance, or don't worry about that one. Whether you will get a certificate and all. Surely we will be conducting a test, and
who are attending in YouTube channel later on, they will also see as a attendant.
Yeah, actually we will be conducting a test at the end of the session. Okay, you please attend the test without missing that. Okay, from that we can consider your application.
Okay, so just I request you to please send me the feedback form for yesterday.
Okay, sure. Okay.
Okay, sir. Thank you so much.
Yeah.
Hello, sir. This is Motesh here, sir.
Yeah, Motesh.
Yesterday I attended class